Say hi to everyone. It's Rimini Margherita from Milan, from the San Raffaele Hospital. In this poster, we presented the results from our cluster analysis on a large cohort of patients with IDH1 mutated intrahepatic cholangiocarcinoma. As you already know, cholangiocarcinoma constitutes a neoplasia with a scarce therapeutic option and dismal prognosis. Moving from the scarce results obtained in a clinical practice with chemotherapy, a new interest in the heterogeneous molecular and genomic landscape of cholangiocarcinoma is currently emerging. The advent of a new innovation in high-throughput molecular and genomic study has recently permitted to highlight new molecular C-target with a potential therapeutic implication in the advanced cholangiocarcinoma setting, including the IDH1 and 2. In physiological condition, IDH enzymes are involved in important metabolic reaction into the cell, and gain-of-function mutations of the IDH genes resulted in DNA and histone hypermethylation, genetic instability, aberrantly expressed hypoxia gene signature, oxidative stress, and altered mitochondrial electron transport change and mTOR pathway. In cholangiocarcinoma setting, IDH1 mutated forms have been highlighted to be exclusively present in intrahepatic cholangiocarcinoma, where represented 25% of the cases, with differences depending on the geographical location. In the last years, many efforts have been made by the research community in order to explore the therapeutic implication of IDH genes mutations. The promising result from the prospective randomized clinical trial Clarity led to the FDA approval of the IDH1 inhibitor ivosidenib as a therapeutic choice for advanced and metastatic cholangiocarcinoma patients harboring IDH1 mutations. Nevertheless, if clinical trials are showing interesting results, it is possible that the high molecular heterogeneity existing in this group of neoplasia could limit the efficacy of ivosidenib in advanced cholangiocarcinoma patients carrying the IDH1 mutations. So the aim of this work was to deepen into the molecular landscape of IDH1 mutated cholangiocarcinoma, thus performing a clustering analysis able to classify patients in different groups characterized by altered genes with a clinical impact and prognostic significance in of patient treated with surgery, and in another one of a patient receiving a first-line systemic treatment. For the sample study, we selected 125 consecutive IDH1 mutated cholangiocarcinoma patients treated for resectable, for locally advanced or metastatic disease in 6 Italian institutions and 1 Spanish institute from January 2013 to March 2021. This sample included the patients diagnosed at the local stage who received radical surgery, patients who relapsed after surgery, and patients diagnosed at locally advanced or metastatic stages without indication to surgery, thus exclusively receiving systemic treatment. All patients were diagnosed as IDH1 mutated intrahepatic cholangiocarcinoma by NGS through the Foundation 1 assay. Genomic data were collected into electronic files by each participating co-investigator in order to perform a mutation-based clustering study with a CCPV model from Zhang and colleagues. This method permits to cluster tumor based on the somatic mutation spectra of the putative neoplasia driver genes. We further extended this study to identify prognostic signatures that are quantified by gene expression levels and characterized by somatic mutation spectra of their genes. Overall, 125 IDH1 mutated cholangiocarcinoma patients were retrospectively analyzed and considered for the survival and clustering study. From the clustering study, 3 main clusters resulted since were characterized by mutations in genes belonging to different pathways. Of note, no significant differences were reported in terms of clinical characteristics between the patients included in the cluster 1, 2, and 3, as we can notice in the first table. The cluster 1 included 44 patients showing a mutation in 64 genes. The most altered C pathways in cluster 1 were cell cycle and apoptosis, RTK-RAS, PI3K, and chromatin modifications. The cluster 2 included 64 patients presenting 42 mutated genes. In this cluster, the most affected pathways were chromatin modification, DNA damage control, PI3K, and RTK-RAS. Finally, the cluster 3 included a small number of patients characterized by mutation in 27 genes. The most altered genes in the cluster 3 were cell cycle and apoptosis, DNA damage control, TP53, and chromatin modification. Of note, for each pathway, the relative altered genes as well as the frequency, cluster 1, 2, and 3 were different. Once I identified the 3 main genomic clusters in our sample, we proposed an easy-to-use algorithm able to stratify patients in this 3 clusters on the basis of the genomic profile as revealed by the Foundation 1 assay. According to the results of our study, we chose the presence of alteration in TP53, ATM, CDKN2A and 2B, ARID1A or PBRM1 or BAP1 or PIK3CA or RAS or APC as points in our algorithm, thus leading to stratify patients in our sample. On the right side, we can observe the results from the survival seq analysis. Firstly, we evaluated the correlates of a genomic cluster with clinical outcomes in patients treated with surgery. As reported in the figure 3A, patients in cluster 2 show a significantly better disease-free survival compared to patients in cluster 3 and 1. In the same way, this patient in cluster 2 shows a significantly better overall survival from surgery compared with in cluster 3 and 1. Then we evaluated the correlates of a genomic cluster with clinical outcomes in patients receiving first-line systemic treatment for advanced disease. In this setting, our analysis show that the patient in cluster 2 experienced a significantly better progression-free survival compared with patients in cluster 1 and 3. Moreover, patients in cluster 2 show a tendency toward a better survival from first-line treatment without reaching a statistical significance. Finally, we evaluated the correlates of a genomic cluster with OS from the time of to first-line systemic treatment. In this setting, patients in cluster 2 show a significantly better overall survival compared with patients in cluster 1 and 3. These data are of particular interest if we think about the recent results from the trials investigating IDH1 inhibitors. From the updated survival results from the Clarity trial, when I used it for crossover, the 50% of patients treated with ivosidenib show a significant benefit in terms of OS when compared to placebo, but no genomic definition about which patients could be more likely to respond to IDH1 inhibitors are already available. The present study has analyzed a large sample of IDH1 mutated cholangiocarcinoma patients who didn't receive ivosidenib or other inhibitor of IDH1, thus highlighting a potential prognostic impact of the identified clusters. In the next future, by testing this clustering classification in a cohort of IDH1 mutated patient treated with ivosidenib, a predictive role could be highlighted, and a better definition of patients who could benefit from IDH1 inhibitors emerged. We performed a comprehensive genomic and analysis on a large sample of IDH1 mutated cholangiocarcinoma patients, thus highlighting the presence of three clusters characterized by distinct gene signatures with a prognostic value and a potential therapeutic implication. In the next future, it will be interesting to investigate the eventual predictive role of this free cluster on a cohort of IDH1 mutated cholangiocarcinoma patients treated with ivosidenib, with the aim to define and predict the subset of patients which are more likely to respond to IDH1 inhibitors. Moreover, this first insight into the molecular heterogeneity of IDH1 mutated cholangiocarcinoma patients could open new research ways on the mechanism of primary resistance to anti-IDH1, with the final aim to explore new combined or sequential therapeutic strategies and to improve the clinical management of IDH1 mutated cholangiocarcinoma patients in an optic of precision medicine.